Despite making great strides in controlling HIV, combination antiretroviral therapy, or CART, is still unable to eradicate latently infected cells, and their presence continues to affect immune reactivity. The inflammation that results is then left to compromise the body's other physiological systems. The innate immune system consists of cells and proteins that are always present and ready to mobilize and fight invading organisms. The adaptive immune system, on the other hand, is called into action against pathogens that are able to evade or overcome innate immune defenses. Both can become dysfunctional in response to HIV infection. Researchers suspect that immune dysfunction, along with other factors such as nutritional, hormonal, or metabolic dysfunctions, can still contribute to HIV-associated wasting in the current era of CART. The innate immune system releases pro-inflammatory cytokines upon first exposure to HIV. The inflammatory process is driven by various cytokines, including tumor necrosis factor alpha, or TNF-alpha, interleukin-1, or IL-1, and interleukin-6, or IL-6. In the presence of these cytokines, a breakdown of protein known as muscle proteolysis occurs. As this response becomes chronic, the continuing breakdown of muscle can lead to loss of lean body mass. Research shows that higher levels of catabolic cytokines TNF-alpha, IL-1, and IL-6 have been associated with loss of lean body mass, and changes in resting energy expenditure in HIV-positive study participants treated with antiretroviral therapy. Adaptive immune activation can also contribute to HIV-associated wasting in patients treated with antiretroviral therapy. A profound loss of adaptive immune system protection can occur with CD4-positive and CD8-positive T-cell depletion and dysfunction. This loss happens alongside an increased resting energy expenditure and increased protein catabolism, accelerating the loss of lean body mass during HIV infection. The dysregulation of the adaptive immune system that occurs with HIV infection may also be associated with bone loss. In HIV-positive patients with osteopenia or osteoporosis, a greater frequency of activated CD4-positive and CD8-positive T cells are found. When virologically suppressed on CART, those with lower bone mineral density had a greater frequency of activated CD4-positive and CD8-positive T cells. This finding suggests that some immune activation leading to decreased bone density occurs, despite virologic suppression. The clinical significance of these findings remains to be determined.